Hello everyone and greetings from the Precision Nanosystems team. Before we begin, I would like to make you aware of our upcoming symposium entitled Nanomedicines, Materials, Manufacturing, and Therapeutic Applications. This event will be taking place in Seattle, Washington on July 16th at the Washington Athletic Club from 8 a.m. to 6 p.m. PDT. We will showcase exciting talks from researchers from both academia and industry spanning several topics. Precision's team of engineers and scientists will be available to demonstrate the NanoAssemblr product suite, including the NanoAssemblr Spark, a new low-volume instrument designed to manufacture nanoparticles at the microliter scale. The NanoAssemblr Benchtop, our current instrument for rapid nanoparticle development at volumes between 1 and 15 milliliters. The NanoAssemblr Blaze, a new preclinical scale-up instrument for batches of 10 milliliters to 1,000 milliliters, with the option of automated dilution post-manufacture. Finally, a continuous flow GMP scale-up system that uses 8 microfluidic mixing chips in parallel to manufacture 25 liters in approximately 4 hours. If you're interested in learning more about this event and registering, you can do so for free at seattlesymposium.eventbrite.ca. If you can't be in Seattle for the 16th but would still like to participate, we'll be hosting a live webcast of the event throughout the day. You can register for that at seattlesymposiumwebcast.eventbrite.ca. Now I'm happy to present microfluidic strategies to improve encapsulation of hydrophobic drugs in nanoparticles. I'm Shyam, Field Applications Scientist at Precision Nanosystems. It's my pleasure to introduce today's speakers. Dr. James Taylor is a co-founder of Precision Nanosystems and has led P&I's technology commercialization team since the technology's invention. He holds a BASc in Engineering Physics from the University of British Columbia and a PhD in Genetics from the Institute of Systems Biology. Dr. Taylor has nearly a decade of experience in microfluidics, nanotechnology, and systems biology. James is also a member of the Board of Directors for Life Science British Columbia. Dr. Shyam Garg is a formulation scientist at Precision Nanosystems and is currently using P&I's technology to develop new applications focused primarily on polymeric delivery systems. Dr. Garg has a bachelor's degree in pharmacy from the University of Mumbai, India, and a PhD in pharmaceutical sciences from the University of Alberta. Shyam is experienced in the field of nanotechnology-based medicines, specializing in polymeric delivery systems. Now, before we begin the webinar, some basic housekeeping. If you experience technical issues, please use the question or chat box, and we will do our best to troubleshoot. If you have any questions for our speakers, please use the same features, and we'll try to answer as many questions as possible at the end of the session. If we can't get to your question, we will answer them via email after the event. Now, without further delay, Dr. Taylor, the floor is yours.
Great. Well, thank you very much, Shel. We look forward to discussing our talk on microfluidic strategies to improve encapsulation of hydrophobic drugs in nanoparticles. Advances in chemistry have led to the discovery of many promising new small molecule drug candidates. However, nearly 90% of these molecules are hydrophobic in nature and hence are poorly water soluble. This has led to their inability to transition easily from discovery to clinic. With this in mind, the focus of research has now shifted towards the development of various drug delivery technologies that can enable these drugs to reach the market. Nanomedicine, or nanoparticle formulation and delivery, is a key technology enabling the administration of many small molecule drugs. Significant challenges exist in the formulation of small molecule drugs into nanomedicines. This includes enabling high loading efficiency of drugs, controlling the release profile of the drug, and controlling particle characteristics pre and post encapsulation. And then, of course, these formulations need to be scaled from benchtop volumes to clinical scales under GMP conditions for testing in clinical trials and to be brought to market. In this webinar, we show improved manufacturing of 3 classes of nanoparticle-based systems that are used for the delivery of hydrophobic small molecules. These include lipid-based delivery systems such as liposomes, which can be used for the delivery of both hydrophilic and hydrophobic drugs, polymer nanoparticles made of polymers such as PLGA, PLA, PCl, as well as block copolymers such as PEG-PLA, have been used for the delivery of hydrophobic drugs. And oil and water emulsion systems like nanoemulsions that are usually used for the delivery of hydrophobic drugs, small molecules, or oils. These are widely used for topical delivery and other applications. Precision Nanosystems' contribution to the field of nanomedicines has been to develop improved technologies for the development and manufacture of nanomedicine systems. Our proprietary continuous flow microfluidic platform enables the development of novel nanomedicines much easier, more reliably, and more flexibly than existing technologies. This is due to taking advantage of fluid dynamics at small scales and principles like laminar fluid flow to allow for the exquisite control of the physical and chemical environments for which the nanoparticle self-assembly and nanoprecipitation reactions occur. We combine the control of microfluidics with our continuous flow micromixers to enable mixing in less than a millisecond and in a reactor volume on the order of tens of nanoliters. At the same time, we're able to maintain flow rates that are industrially relevant, whereby our reactors typically operate between 10 and 20 mils per minute. To manufacture 1 liter at this rate would take just over an hour. Larger scales are achieved through parallelization of the single reactor to achieve higher volume throughput without changing process parameters. This seamless scale-up provides confidence that products made in a research or early development setting can be scaled for clinical testing and use. For example, an 8 times parallelization of the microfluidic process produces approximately 6 liters per hour and will result in 25-liter batch to be able to be produced in a 4-hour time frame or a typical production shift. Great. So moving on here, Shyam, as a formulation scientist looking to develop a nanocarrier for the delivery of hydrophobic drugs, what are some of the points you would need to address during the development of this formulation?
Thanks, James. So like the first thing that I would need to do would be to actually design my nanoparticle characteristics for the desired application. So here, for example, size is an important factor that I would consider in this category. The second thing, I would want to test the encapsulation of my hydrophobic drug of interest and also find strategies to actually increase the encapsulation of the drug. And finally, I would want to maintain my nanoparticle characteristics post-drug encapsulation, and which more often than not does not happen, and we're always left finding solutions to try and address this problem. So in today's talk, what we're going to discuss is we're going to discuss these 3 parameters which are during the formulation process. And we've selected Coumarin 6 as our hydrophobic drug. So now Coumarin 6 is a small molecule with a molecular weight of 350. And we're going to try and address these 3 parameters with examples using PLGA nanoparticles, cholesterol-containing liposomes, and also other systems during this process. So let's now walk through the first strategy, which is to tune our particle characteristics for a given application. So what we're going to be showing you in the next few slides is how microfluidics can actually be used to exploit certain process parameters and optimize your nanoparticle design. Process conditions that can be manipulated would include the total flow rate, which is a proxy for mixing speed in our microfluidic mixers. Next is the aqueous organic flow rate ratio, which dictates how many parts of the aqueous phase which would interact with how many parts of the organic phase during the mixing process. Third is the concentration of the nanoparticle forming material, which can impact the nanoprecipitation process. And fourth is the co-solvent systems, which will change the viscosity and the polarity of the system, which again can impact the nanoprecipitation process. So in this talk, we are going to mainly focus on the aqueous to organic flow rate ratio, the concentration of the nanoparticle forming material, and the co-solvent systems. Now, the aqueous to organic flow rate ratio dictates how many parts of the aqueous phase will interact with how many parts of the organic phase during the mixing process. So in these examples, the nanoparticle forming materials were dissolved in the organic phase, with the aqueous phase being either water or buffer. By having an aqueous phase in excess during this mixing process, the solubility conditions of the nanoparticle forming molecules change faster, and this drives the nanoprecipitation reaction faster. So a flow rate ratio of 1 would indicate one part of the aqueous phase is reacting with one part of the organic phase, whereas that of 3 or 3:1, as shown on the slide here, would indicate 3 parts of the aqueous phase is interacting with one part of the organic phase. Now, as the flow rate ratio increases, the polarity change is much more rapid. And what we find is that different systems behave differently. As you can see in the two graphs here, in the case of PLGA nanoparticles, as the flow rate ratio increases, you see there's an increase in the size. And then whereas in the case of liposomes, you see that the size decreases as you increase the flow rate ratio. Next, we move on to concentration of your nanoparticle forming material. Let's see how that can influence the size of your nanoparticles. And again, what we found is that both materials, that is the polymer systems and the lipid systems, behave differently. In this case, you'll see that as we increase the concentration of our PLGA nanoparticle, as we increase the concentration of our PLGA polymer, you end up actually increasing the size of the nanoparticles all the way from 70 nanometers to up to 200 nanometers. In the case of liposomes, what we found is that increasing the concentration of lipids from 10 to about 75 milligrams per ml did not seem to impact the size of the nanoparticles. We find this difference between the polymer and lipid systems interesting and most likely to do with the different nanoprecipitation kinetics. Now, one noticeable result that came out of this study was the ability to formulate PLGA nanoparticles at sizes below 100 nanometers. So most conventional methods are not able to formulate at these sizes, and actually achieving these sizes of about 70 nanometers actually increases the potential of these polymers to be used in many more applications. Next, let's talk about the ability to manipulate size using different co-solvents and also by changing the total flow rate. For this, we're going to use the example of a block copolymer, that is polyethylene glycol block polylactide, which is PEG-PLA. And now using an emulsion solvent diffusion method, we were able to achieve a size of 100 nanometers, as shown here in the bar in blue. The goal was to transfer this method using microfluidics and still be able to achieve the same size of 100 nanometers. So what we did was we made PEG-PLA nanoparticles via the nanoprecipitation method using microfluidics. The PEG-PLA polymer was dissolved in acetone DMSO mixture at a ratio of 90:10, with water being the other solvent. Now, using these two solvents, we were actually able to achieve a size of about 45 to 35 nanometers, as can be seen in the orange bars here. By changing the flow rate from 2 to 10 ml per minute, we were able to get those sizes between 45 and 35 nanometers. On changing to a more viscous solvent, that is when we move to 100% DMSO, you can see that at the 10 ml per minute flow rate, we were able to get a size of 150 nanometers. Next, when we change this further to a 50 acetone DMSO mixture, we were actually able to get that size down to about 50 nanometers. And then finally, when we actually changed the flow rate to 1 ml per minute, we were able to get that size of 100 nanometers. So normally, in the case of block copolymers, what you will see in the literature is that it's very difficult to control the nanoparticle size. Typically, it's required to modify the polymer block length, and this will impact the particle size, but it will obviously also change the entire system, since now you're modifying your polymer itself. In our example, what we are able to show you is that we can tune the particle characteristics using just process parameters like total flow rate or the co-solvent systems without actually changing the polymer itself. And what we were able to do is that we're able to actually tune that size from 35 nanometers to 150 nanometers and also get the size that we wanted, which was the 100 nanometer size range. So next, we'll discuss on how we can increase the encapsulation of hydrophobic drugs into nanoparticles. Now, previous studies by our collaborators has shown that microfluidics can enable higher entrapment of the hydrophobic drug propofol into liposomes when compared to other methods. So as you can see here in the graph on to your left, using the sonication method, they got an encapsulation of about 15%, and when they moved to the microfluidics method, they got an encapsulation of about 45 to 50%. However, even with that increased encapsulation, they did not change the release kinetics of the drug, and you can see that both sonication method and the microfluidics method shows the same release profile, which you can see to the graph on the right. I encourage you to read this paper from Yvonne Perry's lab, as it's an excellent example of how microfluidics can be used to actually increase the encapsulation of your drug when compared to other methods. So using a similar process, what we did was we used Coumarin 6 as our hydrophobic drug to load into liposomes. Now, Coumarin 6 has a high solubility in DMF, which is dimethylformamide, and hence it was dissolved in DMF at higher concentrations and then added to the lipid mix in ethanol, such that the ethanol to DMF ratio was 90-10. On the other end, we used PBS as our aqueous solution. And Coumarin 6 is a bright yellow laser dye, and as you can see, the picture to your right shows drug-encapsulated liposomes. However, we wanted to increase the encapsulation even further. And what we found was that the quick change in the polarity during mixing led to the precipitation of Coumarin 6, and hence less drug was available for encapsulation into liposomes. A possible solution was to decrease this rapid change in polarity, which would avoid the precipitation of the drug and hence make more available for encapsulation into the liposome. So what our team here tried was to reduce the polarity change by adding DMF to the aqueous phase as well. Since Coumarin 6 is more soluble in DMF, as mentioned in the previous slide, the ethanol to DMF ratio in the organic phase was now modified to 80-20 instead of 90-10, as we showed in the previous experiment. And 20% DMF was added into the aqueous phase as well, as can be seen in the diagram up there. What this did was that it slowed the rapid change in polarity and avoided the precipitation of Coumarin 6, making it available for encapsulation into liposomes. As you can see again here to the picture onto your right, it looks much brighter now as compared to the previous slide. So in the graph below, you'll see how encapsulation efficiency changes from 40 to 60% by modifying certain lipids, and shows similar encapsulations using saturated or unsaturated lipids. This can be seen in the bar B and bar E, where the only thing that we've changed is the DSPC, which is a saturated lipid, and we've changed it to POPC, which is an unsaturated lipid. An interesting thing that we found was the importance of cholesterol in these formulations, as a formulation without cholesterol, as seen in bar D, caused aggregation of the particles. This implies the importance of cholesterol in the encapsulation of hydrophobic molecules in liposomes containing saturated lipids, since cholesterol imparts a certain amount of fluidity to the lipid bilayer. Based on our findings, by making these changes, we were actually able to accommodate 10 times more drug into liposomes when compared with previous reports in literature. Okay, so I've done my rational design with my nanoparticles, so I know the characteristics that I want. I've actually achieved them, and I've even been able to encapsulate the drug into this and was able to achieve a higher encapsulation. But now when I'm trying to do my size study, my nanoparticles show a different size. Now this is a common problem. Since most APIs are very expensive, doing a rational design with your encapsulated drug can actually be very difficult. However, what many researchers find out is that once you encapsulate your drug, you're actually changing the nanoparticle characteristics post-encapsulation. So here what we're going to describe to you is how microfluidics can actually be used to maintain the nanoparticle characteristics even post-drug encapsulation. So the first example where we're going to show you is that we saw no change in the nanoparticle size after encapsulation of Coumarin 6 into PLGA nanoparticles. So what you see here is you see the orange bars to the graph on your left. You'll see that empty PLGA nanoparticles show a size of about 75 to 85 nanometers. And then when we load that with the drug, you see that the size does not change and it's still within the range of 75 to 85 nanometers. Again, having these sizes as low as 75 nanometers is usually very difficult in the case of PLGA nanoparticles. And what we did was that using this microfluidic technology, we were able to incorporate the Coumarin 6 drug into these nanoparticles and still not change their sizes. The amount of drug encapsulated, which is about 65 to 75%, as can be seen on the graph to your right, is comparable to what's seen in the literature. However, the sizes we received, the sizes that we achieved were about 75 to 85 nanometers, as compared to the literature in which the sizes they achieved were about 160 to 170 nanometers. The other thing that we compared here was the total flow rate on the encapsulation efficiency. And as you can see to your graph on your right, we also found that change in this total flow rate did not have a big influence on the encapsulation efficiency. Now let's see an alternate scenario where on encapsulation of your drug of interest, the size of your nanoparticles increases. Now this is a more common scenario seen with most researchers, is that as you actually encapsulate your drug, you actually increase the size of your nanoparticle system. So here we have a PEG PLA system, as we had shown previously, and you can see the bar in the blue is for empty PEG PLA nanoparticles, which shows a size of about 100 nanometers. On encapsulation of the drug using the same total flow rate, that is at 1 ml per minute, the size bumps up to about 230 nanometers. But what we can do here is, by just making small adjustments in the process parameters like the total flow rate, we're actually able to get that size back down to the 100 nanometer size. And as you can see here, with the speed of about 5 ml per minute, we were able to achieve that size of 100 nanometers. And during this whole process, the drug encapsulation did not change.
Great. Well, thank you very much, Shyam. That was excellent. In the last 30 to 40 minutes, what we showed you is how our microfluidic technology can assist you in developing your formulations and help find solutions to some of the problems encountered during the development of nanoparticle formulation. So we went through and demonstrated rationally changing nanoparticle size for the required application, increasing encapsulation of hydrophobic drugs into nanoparticles, controlling the encapsulation of therapeutics by exploiting process parameters, and maintaining nanoparticle characteristics pre and post encapsulation of hydrophobic drugs. And that concludes our webinar. I hope you enjoyed it. And here is Shyam.
Thank you so much for joining us today. We do have a couple of questions from members of the audience. The first question is about the cholesterol, and it reads, how would cholesterol impact the release profile of drugs from liposomes?
So although we did not carry out any release studies with the cholesterol, if you look at the literature, it suggests that it's more of a drug-dependent release profile, where some papers have shown that you'd have a high release with high amount of cholesterol, and some others have shown that you'll have a slower release with a higher amount of cholesterol. However, we feel that if the drug was to release purely by the diffusion process, we would think that in the case of an unsaturated lipid, having more cholesterol would actually decrease the release of the drug, since I think it would impart some amount of stability to the membrane. While in the case of, I guess, saturated lipid, we would guess it would be the exact opposite, since it would introduce gaps into the lipid bilayer.
Thank you, Shyam. The next question is the co-solvent approach always necessary for block copolymers? Are there any other advantages other than size?
So, no, the co-solvent approach would not always be necessary. I think it would depend on the size that you'd want finally for your nanoparticles. So, for example, if I wanted a size of, say, 35 to 40 nanometers, then maybe I wouldn't need I wouldn't need the co-solvent system. It's only when I wanted the higher sizes, which when I required to use the co-solvent. and then previous studies by some of the other users have shown that actually it depends a lot on the blocks that you're using. So it's specifically based on the molecular weight of your PEG block. So if you have, say, for example, a PEG 2000, then it's going to show a huge change just by changing, say, the total flow rate. But in our case, where we had a PEG 5000, we actually were getting sizes about 35 to like 40 nanometers when we were only using acetone as our solvent, and hence we had to use the co-solvent to actually enable us to increase that size. The other factors which I think it would affect other than size would be the encapsulation and could also affect the release. However, in our case, we did not see that happening. The other important thing is to think about scale up. So say I have, I'm doing the formulation in acetone and I'm getting a size of 50 nanometers, but I need the flow rate to be at 1 ml per minute. But then I add acetone and I've added DMSO at a, say, 50-50, and I'm getting the same 50 nanometers, but at 10 ml per minute. So when I am to scale up that to say about, for example, a 200 ml formulation, the 1 ml per minute would take about 200 minutes, but the 10 ml per minute would take about 20 minutes. So, as you scale up your formulation, that gap is just going to get much bigger. So yeah, that's mainly, I guess, the other advantage that's there.
Thank you, Shyam. I think that's all the time we have today for questions. If we didn't get to your question, we will answer them via email after the event. If you have any questions about the technology or would like to learn more about the NanoAssemblr, please visit precisionnanosystems.com. And don't forget to follow us on LinkedIn and Twitter. From Dr. Taylor, Dr. Garg, and myself, have a wonderful rest of your day. Cheers.